Sure, because I haven't had a chance to meet everyone in this meeting, I'm going to talk a little bit about my background for the first two slides. So, in my academic background, I'm currently in the fifth year of a Bachelor of Science in Electrical Engineering co-op, the Nano Engineering option here at the University of Alberta. And I have some previous electrical engineering work experience with power systems with the City of Edmonton's LRT system and Suncor's oil sands extraction. And I also have previous academic experience in some nano engineering courses, including a nano electronics course and this course, Analytical Methods in Electrical Engineering, which is more like a mathematical course that kind of gives me the background for the physics that goes on a nano. And also, in case some people, have had the chance to meet me and they might not be able to, because unfortunately I'm here for the summer. So just a bit more background on me. I'm from Serbia. I moved to Canada when I was young. I grew up in Fort McMurray, so that's a town that's north of here by 5 hours, and came here to Edmonton for my undergraduate studies. In more of my free time, I'm a student member of two engineering organizations, Society of Photo-Optical and Instrumentation Engineers, a Canadian Nuclear Society, and over the summer so far, I've been enjoying playing soccer and beach volleyball. So, okay, into my summer project. It's on AFM, which stands for Atomic Force Microscopy. And it is a form of microscopy that uses a sharp tip or a probe to scan the surface and to gather information at the nanoscale, similar to kind of what the GIF is showing. And this technique can produce high-resolution images uh, of surfaces at the nanoscale, and can also give us different types of information about some of the properties of the sample. And what's important to note here is that it does not use light. Usually microscopes are optical microscopes, and they have a system of lenses to magnify light. This one relies on a sharp probe to get all the details of what's going on the surface. Now, a little bit more detail as to how it works. First, the tip is brought very close to the surface, and then as the tip kind of scans across the surface, the forces, which at this small scale are intermolecular forces, cause the tip cause the tip to move up and down, and the cantilever in the diagram that's attached to the tip to move up and down. And as you can see in the picture, that there's a laser beam kind of reflecting off the top of the cantilever. So as the cantilever moves up and down, the position of the reflected laser will move on the photodetector. And this is how the change in deflection of the cantilever is measured. And then this deflection of the cantilever, this data, is used to construct a high-resolution topographical map of the sample surface. So it's used to generate an image of what the probe is seeing on the sample surface. Now, that is the basic physics of how it works, but there's also two different modes that I should probably go over briefly. First would be contact mode, which, as it suggests, the probing tip is in physical contact with the surface throughout the entire imaging process. Now, the advantages of this imaging process, this imaging mode, is that it's pretty high resolution imaging, but there is a slight risk of damage to either the cantilever tip or to the surface. So what we've been mainly focusing on for my project is the second mode, which is non-contact mode. So in non-contact mode, or as it's also called, vibrating mode, the probe at the end of the cantilever is vibrated up and down. And then as the vibrated probe, oscillating probe, interacts with the surface, the amplitude of its oscillation is dampened. And then this information is fed back to the electronics controlling the cantilever and the probe. And then it's adjusted to keep its amplitude the same value. So it can be adjusted up or down. And as it's getting this feedback information through this feedback loop, it's using that to generate the topographical image. And the amount of force that is between the sample and the tip is reflected in the change in the amplitude of the vibrating probe. And then this information is used to make the image. And one of the advantages of non-contact mode, as I was mentioning, was that it's a non-invasive mode. So it's not going to really damage the sample or the tip. And it's very useful for imaging delicate samples like soft biomaterials, which I'll kind of expand on a little later in the slides. And there's also one kind of a subset of non-contact mode I wanted to kind of focus in a little bit more on, which is called phase imaging. So the image down here actually kind of shows some of the unprocessed images and results I was getting as I was using an AFM. And as you can see, there are three pictures in the bottom image, three different graphs being generated. The first one is kind of like a height map, the second one is a phase map, and the third one is the amplitude map. Now, the middle one that we're going to focus on the phase map is particularly interesting because how the phase imaging works is that there's a signal that's driving the oscillation of the cantilever, and then there's the actual oscillation of the cantilever. And if you can imagine, if the tip is oscillating in kind of open space or on a completely uniform surface, there will be no difference between the signal that's driving the movement and then the actual movement. But on other surfaces that are not uniform, if there's a difference in the mechanical properties of different regions of that surface, different regions are harder or rougher or softer, then uh, then the actual oscillation of the cantilever won't match the driving signal, and this phase difference is captured in phase imaging and used to generate the image. And so this is particularly useful to get the mechanical properties of the surface, such as friction or adhesion, which is the attractive force between the tip and the sample, and also viscoelasticity, which just means how the surface deforms, is pressure too hard, and how it recovers from stress as it's relaxed a bit. And also, we're going to talk about some of the capabilities of AFM, kind of what it can do. So the first one, the most obvious one is to actually generate the images. So to provide 3D images of the surface topography, so you can see what's on the surface at this very small scale, with resolution that's atomic or near atomic resolution. And even based off these images, you can get additional analysis, like the roughness of the surface or the texture of the surface. But I also want to talk a little bit about something else you can use AFM with. It's called force spectroscopy. I didn't use it in my project, but I think it's a very important one to talk a little bit about. In force spectroscopy, kind of how the image shows, you get the AFM tip and you move it towards the sample, which is called the approach phase, which actually is moved all the way to cause some kind of deformation in the sample. And then it's retracted during the retraction phase. Then the sample returns to its original shape. And the data you get from this are force distance curves, just kind of graphs, which provide information on adhesion, which are the attractive forces, and some of the elastic properties. So the ability for the sample to kind of uh, recover from the deformation and return to its original shape and size. And one of the kind of standard ways of using force spectroscopy is through a method called nanoindentation, which is used, just as I mentioned, to measure sample's hardness. There's even something called an elastic modulus you can use to rate different materials and other sort of deformation properties that are useful in the particular research, if it is. And even though the method of AFM I was using for my project was kind of the non-contact mode, as I discussed earlier, I'm going to kind of quickly go over kind of different versions of AFM you can use. There are kind of different variations that have different purposes. I'm going to quickly go over kind of how they work and kind of basic applications. So one of the first ones to talk about, I guess, would be conductive AFM, which kind of how the picture shows up there. It's simple, a voltage is applied between the conductive tip and the sample. And then, same as before, the tip scans across the sample. And not only do you get a topography image, but you can also see kind of the electrical conductivity of different areas of the sample because the current is also constantly measured as it scans across the surface. So you can see the electrical properties of the sample as it moves across. And another one also quickly to mention is electric force microscopy, which is used to detect electrical forces in addition to topographical imaging. And the way it works is, to maybe explain the image a little bit, is the first time the probe scans across the surface, there are no electrical forces, there's no charge on the tip. So it just gets the kind of the topography of the image, knows what all the hills and valleys are. Then the second time it scans over it, the tip is then charged with an electrostatic force, and the sample, for example, has other electrostatic forces there. So because the tip already went the first time and it knows where all the hills and valleys are, it retraces the same path. But if it feels additional forces along this path, then it can use that to map where the electrical forces are along the sample, which is very similar to this next kind I want to talk about, magnetic force microscopy. It's very similar to electric force. The first time they just use a regular tip to go through the topography of the sample. And the second time they use a magnetized tip in order to see the different magnetic forces that are on the sample that they're interested in measuring. And then another one that's also kind of interesting is scanning thermal microscopy, which is similar idea as before. The probes are scanning through the sample. Only this time, the probe is very specialized and equipped with a thermal sensor, usually a thermocouple that at different temperature areas on the sample, it will react with a different mechanical movement, which then makes a different current, a different signal, and then through that it can measure different temperature changes within the sample and kind of get a map of the temperatures on the sample, kind of like the image to the right with the yellow and purple. And then these last two kinds I wanted to talk about, which are kind of very interesting too. The first one is chemical force microscopy. So very similar to the picture that's shown there. You have the tip that's actually functionalized, which means it's coated with specific chemical groups. And then CFM measures the chemical interaction between the tip and the sample. And the way it does that by bringing the tip into contact with the sample surface, seeing the force required for the attraction, and then bringing it away, seeing the force required for the separation. And that way they get some information about the chemical interaction between the tip and the sample. And the last one to quickly talk about is infrared AFM, which very similar to other AFM, except the sample is first eliminated with infrared light, which is absorbed by very specific chemical bonds that absorb energy and vibrate. And then as the tip interacts with the sample, it can detect these areas of surface vibrations. And basically, not only do you get a topographical map, but you can also get some information on the chemical composition of certain parts of the map, some of the orientation, some of the other properties of the material. So I want to discuss uh, now the applications of atomic force microscopy in research, and we're going to focus on life sciences. So the main way it's useful here is AFM allows the nanoscale imaging of some soft biomaterials, including cells, DNA, and other biomolecules in both regular atmospheric conditions, as well as liquid environments, which is very interesting. The first example, like the image to the left, is DNA molecules. This is 5 micrometer by 5 micrometer image of double-stranded DNA molecules. And imaging these short sequences of DNA, such as these, can be useful in the study of biological macromolecules or in the study of interacting these DNA strands with other molecules like enzymes. And then here's an interesting one, epithelial cells. This is an image of one of the outer cells on skin, on blood vessels and organs. And what's interesting about this is it's measured in liquid. So AFM has the ability to do high resolution images of cells in liquid, and you can get the liquid to kind of mimic realistic physiological conditions that are found in organisms, which is another possibility unique to AFM. And then also on the bottom left, we have a 1.2 micrometer image of the tobacco mosaic virus. So it's also possible to imaging viruses, also in the study of other biological molecules. So here on the top left, we have image of bacteria spore mutants. So AFM can also be used to image a large number of cells, bigger images such as these spores. So the information this allows the researcher to obtain important information about a whole group of cells. And you can also learn things about the interaction of cells, such as clustering as cells form groups and adhesion as cells stick together. And then on the bottom, these two images are very interesting, even though they didn't come from me using the AFM, they were kind of very similar. Because in the bottom middle image, you can see that that first image in the bottom middle actually comes from an optical microscope, where they're kind of getting a bigger picture of what's on the sample. And that kind of shadowy figure to the right in the middle image is the actual probe, the cantilever tip. And then once you kind of select an area that you want to image with the optical microscope, like for example, in the middle picture, they have a black box in the middle left. They've kind of selected that area that they want to image with the actual AFM. You then kind of move the cantilever, which is just that shadowy area in the middle, into that area, and then it kind of gets a more magnified image of what's going on in that very small area of the optical microscope. And in this case, the images I found were of imaging white blood cells, first with the optical microscope on the left, and then zooming in more and getting a more detailed image with the actual atomic force microscope of the same white blood cells in the photo. Now, kind of bring it back to my work over the summer and to Wishart Lab. Kind of my goal for the summer was eventually to produce an image of this PTCDA molecule to see its chemical structure. And to do that, it's required to first kind of be absorbed onto this silicon surface into one of the corner holes on the silicon substrate, and to do this imaging at room temperature. And uh, and eventually to apply this imaging technique to the metabolites that the team is currently studying. Now, as I'm going to kind of discuss in the next two slides, that to actually image a single molecule requires kind of a very sensitive atomic force microscope with very good resolution. For example, the PTCDA molecule, its dimensions are roughly 1 nanometer, 1 nanometer, 1 nanometer. So we would need to get an atomic force microscope with a Z resolution of around 1 nanometer to successfully measure it. And even to apply it to the other metabolites the team is currently studying, most metabolites are also very small, around that same size, from around 1 to 10 nanometers in dimension. So that is kind of one of the challenges coming up. And here I have a couple of pictures of the results that I've been able to kind of collect a couple of weeks ago using one of the atomic force microscopes in the chemistry center sub-basement. To kind of test out this atomic force microscope, we were imaging graphene samples to kind of see uniform layers of graphene, which is kind of what we're seeing here. In the images I should probably describe, we start with kind of really big lateral sizes, 2 micrometers by 2 micrometers. We move to 1 micrometer by 1 micrometer, and we slowly get smaller lateral sizes. And then kind of when we get 1 meters is when noise kind of takes over and we're unable to see very, very good images of the graphene grains. But yeah, what's important to note here is that to the right of each of the images, each of the 6 images, there's a bit of a scale. I don't know if you can see my cursor to the right, where it goes from dark is around -6 nanometers to the bright part, which is about +5 nanometers, which means we have a scale of around 11 nanometers in the Z resolution, because X and Y is kind of the other measurements. And like I mentioned earlier about the actual size of the ideal molecule we're trying to image, the PTCDA molecule, which is more around 1 nanometer-ish, uh, its vertical height. And right now we're at about 11-ish, 10 nanometers. So one of the goals moving forward is to kind of improve resolution in the Z direction in order to be able to succeed in our goal into able to image the PTCDA molecule. And then, yes, and then here are some ideas we've discussed about when it comes to improving resolution in order to kind of be able to image very small molecules. It's basically one of the ideas is to limit noise in the Z direction, and then there are many forms of noise. The first one is acoustic noise, sound waves. So there are a couple ideas to limit sound that can interfere with the AFM, especially because we're in vibrating mode. So any kind of noise, it's a little bit more prone to affect the measurements. So to stop acoustic noise, maybe we can use either some kind of enclosure on the AFM that blocks sound, or to kind of put it in a room just with good soundproofing, going to basement away from other things that are noisy. Also, another big consideration is vibrational noise. Kind of how in the 8th floor there's the construction that goes on that vibrates the whole building, but even smaller things like the air conditioning in the rooms can have vibration that affects it. And one of the better strategies to dealing with this is an anti-vibration table that's able to kind of level out all those vibrations and keep things precise. And also electrical noise, because the atomic force microscope, it's all electronics to kind of control everything. The scanner, the probe, and everything is in a feedback loop. So to limit electrical noise, an idea for that is to properly ground all the equipment and to use a common ground for everything to make sure there's no discrepancies to kind of limit electrical noise. And then also other ideas we have moving forward to improve resolution is to adjust scan parameters. So these are the actual settings in the nanoscope software we use to control the AFM. Things from scan rate to take longer to do every scan to get more of the details finely tuned, to get Z range accurately measured also in the settings and gain settings. And then also we reached out to the company that was able to that actually built the microscopes we were using, and they confirmed that it's possible to achieve a sub-nanometer resolution with their equipment. So that is one of the goals, to eventually limit noise and stuff like that, to be able to image the PTCA molecule on a very small scale. And for the last couple of slides of the presentation, I kind of wanted to highlight where kind of atomic force microscopy and metabolomics kind of intersect, and kind of applications we can use atomic force microscopy to help with different areas of metabolomics research. And so I kind of picked the different examples from papers I found online that seemed very interesting to me. And so one of the first ones I'll start with is to be able to discover disease biomarkers earlier than usual. So to do an early diagnosis of disease is challenging with kind of regular instruments, but atomic force microscopy is very precise on a nanometer level that it can detect very small changes in either cell shape or metabolomic profile that can lead to an early diagnosis of a disease. So uh, a paper by Dr. Eaton of the University of Lisbon. They utilized atomic force microscopy to observe the morphological changes in a type of white blood cell in acute asthma patients. So in the pictures below, to the left is the white blood cell from a healthy individual imaged by AFM, where it's very round, very complete. White blood cell seems very well formed. And then to the right is an image of the same white blood cell, but with an individual who has the asthma symptoms. So here you can see the white blood cell. It's regularly shaped, it's deformed, doesn't look proper. And when they were conducting the study, they had prior knowledge that changes in these types of white blood cells correspond to the progression of inflammatory diseases. So they were able to use the precision of an AFM to be able to see these changes way before they're usually observed through like bigger changes in someone's body and stuff like that. And then kind of jumping over to kind of a different area, metabolomics with pharmacology. Here's an interesting example of drugs being kind of investigated and with AFM. So in this particular example, this paper I found by Dr. Ratha, they were treating diabetic patients, which they used insulin to treat diabetic patients. And insulin is a protein hormone. And the problem with it is insulin can kind of fold the wrong way and lose its proper structure. And then once it loses its structure, it can't perform its function to treat diabetic patients. And then what's worse is these insulin proteins that are folded the wrong way and lose the structure, they can kind of group up and form these things called fibrils, which can then cause tissue damage and all kinds of other problems. So in this paper, Dr. Ratha investigated potential drugs that can stop the fibrillation process in insulin, stop it from grouping up and losing its proper structure. So using these pictures below, on the left is the starting sample they had of insulin when they first put it together. As you can see, everything is kind of uniform, lots of small grains. Everything kind of looks normal. And then they had two different groups of these insulin proteins. In the first group was the control group, where they just simulated conditions that are favorable for it to form these groups and lose their structure. And that's what the middle picture is showing. A couple hours in, with the control group with no drug kind of formed the fibrils. And as you can see, they're very dense. Uh, they kind of combined and grouped up, and you can see them in the AFM image pretty well. But meanwhile, the picture on the right is the group that had a peptide molecule. So it's this kind of drug they're kind of investigating to see if it works. And it kind of affected the insulin and kind of made everything spread out more and not group up and clump together. So it was successful in stopping the process of fibrillation with one of these drugs. Another interesting example, I thought, if we now kind of move to plant sciences a little bit, was a paper that was kind of focusing on preserving food in different methods, again, kind of investigating drug treatments. So here, Dr. Chen studied the effect of ATP treatment on the preservation of mung bean sprouts. So it was a type of food they chose, these mung bean sprouts, to see if they can make them last longer. And one of them they treated with ATP treatment, and one of them they had a control group. And what they used the atomic force microscopy for was to image the cell walls on the actual mung bean sprouts. And they were specifically focusing on one kind of molecule that we can see in the bottom left image called the pectin molecule, which is in all the cell walls of all kinds of fruits, not just mung bean sprouts. And they were investigating the idea that all these thick branches you see on the bottom left image of the pectin molecule, that is for the control group at the start when the food is fresh. And then on the third day with no ATP treatment, the pectin molecule kind of degrades. There's no more branches. And in the AFM image, there's just one little, in the middle picture, you could just see one little branch, but really most of the molecule is gone. And then this corresponded with bigger changes in the actual mung bean sprout of getting brown and rotting and things like that. But meanwhile, also on day 3, they used an ATP treatment. So they treated one of the mung bean sprouts with ATP. and they saw that lots of the pectin molecules in the cell wall that they imaged still had lots of the branches kind of still alive, still maintained, and that corresponded also with the mung bean sprouts not rotting so fast, kind of maintaining its integrity and firmness. And they used AFM to get the image of the cell wall to see this pectin molecule, and that's another unique possibility with AFM. And here, as the last example, which I thought was very interesting, was I found a study where they were studying the aging process. So here in Dr. Esmond of the University of London, uh, they were studying these small worm-like organisms called elegans, and they knew kind of these small organisms, if they changed certain factors, they could extend their lifespan. So if they changed the temperature where they lived, or they changed their diets, or they modified certain genes, they could extend their lifespan to 2 times longer they live, or 3 times longer, 4 times longer, stuff like that. What they wanted to measure was the health span of the organism. And health span kind of means the strength of the organism, its quality of life, if it's frail or if it can like resist its environment and it's kind of stronger. And they used AFM in order to kind of measure this health span. It's kind of vaguely defined. And they used it to image the cuticle, which is the outer layer of this microscopic organism. And they used AFM in two ways to kind of determine health span. The first way was kind of through force spectroscopy to kind of push very hard down on its on the outer layer and then pull it back up to see its reaction to deforming and then returning to its shape, to see the hardness and the stiffness of the of the microorganism's skin. And I didn't include that data because it's mainly just force curves and it's not super interesting. But the second way they used AFM to kind of measure the health span of the organism is also to kind of image the topography of the outer layer of this microscopic organism, to kind of see kind of what its outer layer of skin looks like. And on the image on the left is kind of a young, healthy, strong health span microorganism that is 1 day old. As you can see on its skin, it's formed very nicely. It's very straight and then goes down straight again. Very kind of uniform shape. But then one of the older ones, near the end of its lifespan, and also with a very weak health span, this is kind of what happens to the outer layer. It gets all rough and misshapen and non-uniform anymore. And these are one of the ways that they used AFM to kind of measure the health span within an organism that they were extending the lifespan of, which I thought was very interesting also, an application. And I think that is the last slide. Thanks everyone for listening. Hopefully I talked a little bit about kind of what I'm doing over the summer and hopefully some applications of AFM in different areas of metabolomics that can be helpful to everyone here at Bushwick Research Group. So if anyone has any questions or any comments or anything like that, feel free to ask them.